So in this video, we're going to talk about carbohydrates, right? Who doesn't like about carbs, right? As when you hear the word carbs, the first thing that's going to come into your mind is going to be like bread, rice, or noodles, right? But it's actually more than that. So first, sugar, right? The most common example of carbohydrates. Sugar, is it just biological energy? Well, sugar is often associated with being bad for health, but it's actually a vital component in biology for storing and providing energy. And it's actually doing a lot more than just biological energy. So, the sugar as being one example of carbohydrates, there's actually one of the four major types of organic compounds. The others being lipid, nucleic acid, and protein. So carbohydrates being the primary source of sugar, right, are organic compounds containing carbon, hydrogen, and oxygen. As you can see in this picture right here, so this is a ring shape of a glucose unit, right? Glucose, when you see the word that ends in OSE, you know that this is a sugar. Glucose is actually the most common type of sugar, and all simple sugar like this can exist in the form of a linear shape or a ring shape. So what you're seeing here is a ring shape of sugar or glucose. You can see that it's made out full of hydrogen, carbon, and oxygen. You can also see the functional group, the hydroxyl group right here. Okay. So carbohydrates not only exist in ring shape, but they also exist in what we call a single sugar molecules, also known as monosaccharide. Or chain of sugar molecules, which we call polysaccharide. Okay. So let's focus on monosaccharide first. Mono means one, and saccharide means sugar, right? Monosaccharides being the monomer of carbohydrates. Monosaccharides has a general formula CH2, and this is multiplied by N, which means the number of carbon, hydrogen, and oxygen follows the ratio of 1 to 2 to 1. So if that particular carbohydrate has 5 carbon, it will have 10 hydrogen and 5 oxygen. And that being said, that number is 5 for that particular carbohydrates. For example, the most common sugar, glucose, is C6H12O6. You're expected to remember the chemical formula of glucose. Different types of monosaccharides can actually have different ways of arranging their atoms, leading to distinct properties. So example right here, we have 4 different types of monosaccharides. If you check it, they all have 6 carbons, right? For example, alpha D glucose has 1, 2, 3, 4, 5, 6, 6 carbon. And then alpha D mannose also has 6 carbon. D galactose has 6 carbon. D fructose has 6 carbon. However, you can see that the way how they arrange their hydroxyl group is slightly different. Alpha D glucose has hydroxyl group at carbon 2 in the right side, and alpha D mannose will have the hydroxyl group at carbon 2 on the left side. And some of them will have some minor changes, right? For example, D glucose and D galactose, like carbon 4 is the one that's different, even though it's really just like left-right difference, right? One hydroxyl group is on the right side, another hydroxyl group is on the left side. That small difference can actually affect their property and their function. So monosaccharides can also be categorized by the number of carbon atoms that they contain. So for example, triose means that it will have 3 carbon, tetrose means it has 4 carbon, pentose means 5 carbon, hexose means 6 carbon, and so on and so forth. Common monosaccharides include glucose, galactose, fructose, ribose, and deoxyribose. These are the more common ones, which we will see in the later chapter. They are all monosaccharides, which means they are the monomer of carbohydrates. Next, Disaccharide. So if you want to make a disaccharide, di means two, saccharide means sugar, you need to have two monosaccharides. So when two monosaccharides bond together through the process of dehydration synthesis, they form a disaccharide. So disaccharides, for example, such as lactose, fructose, sorry, not fructose, sucrose, and maltose. They are the more common type of disaccharides that we will see in AP biology. So they are simple sugars and they provide energy to living cells. But they are made of two monosaccharides bonded together. The connecting bonds in sugar between the monomers are actually called glycosidic bond. Let's look at this particular model right here. You can see the ring shape of the two monosaccharide, and they're being connected by this central bond right here. And this particular bond is called glycosidic bond. Sometimes we'll use the word linkage, but they are synonymous. So in this picture right here, you can see this is a sucrose, right? Combined by the glycosidic linkage, which is made of, and then this particular disaccharide is made of two monosaccharides. One is glucose, one is fructose, and together they form sucrose. Next, oligosaccharides. So oligosaccharides are saccharide polymers with a small number of monosaccharides. It ranges between 2 to 10 units. So they're short, they're not as big as polysaccharide, but they still have important functions. One of them being able to perform cell recognition. So they can be found in the plasma membrane or cell membrane of animal cells. Oligosaccharides can also be attached to proteins, which we call glycoprotein, or lipid, which we call glycolipid. Example of oligosaccharides is basically AB or blood type. So if we look at this picture right here, we can see there's three oligosaccharides that are being attached to the lipid. This is found in cell membrane. You can see the reasons why people have different types of blood type is really because of this oligosaccharide. They're slightly different between people with type A and type B blood. So type A has a very specific type of monomer that's found within its own oligosaccharide as compared to B. And type O doesn't have this particular monosaccharides. They're all oligosaccharides, it's just that the length of their oligosaccharide is slightly different and the composition of their oligosaccharide is also slightly different. Okay. Next, polysaccharides. So this is kind of like the more important parts of this video. So polysaccharides are long carbohydrates molecule formed by repeating monomer units joined by the covalent bond called glycosidic bond. So you have four types of polysaccharides that you need to remember. So the first one is a type of polysaccharides that's found within cell wall in plant cell, and its main function is to provide structural support. This polysaccharide is called cellulose. As you can see there on the picture on the right side, right? It's made of is a picture of many plant cells. The green boundaries is representing the cell wall, which are made of a particular type of carbohydrates called polysaccharide, called cellulose. Next, we have this particular polysaccharide is found in plants and is composed of amylose and amylopectin. This polysaccharide is called starch. This is in cornstarch, right? It's basically from corn. And this particular polysaccharide is acting as a storage of energy. That's why it's very tasty. Next, we have a type of polysaccharide, also known as animal starch. So this is basically storage of energy that are found in animal cells. So they are called glycogen. Another type of polysaccharides can be found in the exoskeletons of certain animals like insects or invertebrates. This particular type of polysaccharide is called chitin. Since they are all polysaccharides, they are made of the monomers monosaccharides. If you use the process of hydrolysis, you'll be able to release monosaccharides from polysaccharides. Vice versa is also true. Right? You can use dehydration synthesis to take monosaccharide and stitch all these monosaccharides together to form polysaccharide. So this is the picture of the polysaccharides at the molecular level, right? So you can see that even though these three polysaccharides are made of the glucose monomer, the way how they arrange within itself is slightly different. And this contributes to the differences in their properties. So you can see that we're going to look at cellulose first. You can see that cellulose is able to form lots of hydrogen bond within the glucose repeating units inside cellulose. So remember, hydrogen bond is actually a type of attraction between the molecules. So because of the presence of lots of hydrogen bond in cellulose, this makes cellulose really, really strong and rigid. So it means it's not easily broken down. That's why cellulose is used as the structural support of plant cell. Whereas glycogen and starch, you can see that they don't really have that many hydrogen bond and it's spiraling, right? So what it means is that enzyme is easier to break all these monomers of glucose and release them into individual glucose. And we can use this glucose as a source of energy. So when we don't need that many glucose, we can do the opposite, right? We can stitch these glucose subunits and put it into the polysaccharides, making it a storage of energy. So next, we're going to talk a little bit about enzyme specificity. We'll talk more about enzymes in the future chapter, but basically enzymes is a molecule that can speed up chemical reactions. So the idea is that because these polysaccharides and disaccharides, they have very specific type of bond because their monomer is slightly different in the shape. As a result, they have very specific way of binding to these polysaccharides and carbohydrates in general. So specific enzymes are going to bind to a specific type of polysaccharide. So here, because grass are very rich in cellulose or leaves, very rich in cellulose, our body cannot break down this particular type of bond that are found in cell wall. So as a result, we cannot really get a lot of nutrients and energy from these plants. However, for example, sorry, I went too far. So because of all these cellulose are unable to be digested by our body, we don't have that type of enzymes. So all this insoluble cellulose are going to pass through our digestive tract as insoluble fiber. Insoluble means it cannot be dissolved in water. So it's going to help with our digestions. Some microbes are able to use enzymes to digest cellulose, as found in this picture of a cow. I believe the enzyme is found here. Might be wrong. So many herbivores from cow to termites have this symbiotic relationship with these microbes that contains these particular enzymes that can break down cellulose found in plant cells. As a result, the cow and herbivore are able to extract the energy from this relationship. Okay, so in summary, carbohydrates, source of energy, but can also be used as a structural support. Monomers are monosaccharide, polymers are polysaccharides. You're expected to know different types of polysaccharides, but they're all made of the same. They're all made of monomers, carbohydrate, monosaccharides, but they can be of different types of monosaccharides. And remember the relationship between dehydration synthesis and hydrolysis. Dehydration synthesis, we put things together in order to build something bigger, right? We're putting the monosaccharides together to make a bigger polysaccharide. Hydrolysis is the reverse. We're using water molecule to break the polysaccharides into monosaccharides. And remember the main idea here, structure determines function. Because the monosaccharide has slightly different shape, as a result, it will form slightly different bonds between the monosaccharides. As a result, it will make their function slightly different if you build it into polysaccharide. That's it for today.